for this morning. Another new addition to SEBA, but not a new graduate as well. Dr. Parisa has been working in Malaysia in UPM for several years before she joined us in SEBA on 31st December last year. So she's just been with us a short while. And so today we would like to get to know her and her research better. Her research interests range from plant genetics, molecular biology, and breeding to whole crop physiology and the use of molecular marker technology for genetic improvement of plants. And she's especially interested in underutilized crop species. So I invite now Dr. Parisa to present her talk on overexpression of the Pikh gene leading to improved blast disease tolerance in rice. Welcome, Dr. Parisa.
Please turn on your microphone, Dr. Parisa.
Good morning, everyone. Thank you, Prof. Jenny, for introduction, and thank you all for being with us. Actually, I have a bit stress because I don't have any experience on using Google Classroom. Okay. As Prof. Jenny introduced me, I'm Parisa Azizi, graduated from UPM. I would like to talk about some part of my PhD thesis entitled Overexpression of Pikh gene leads to improved blast disease tolerance in rice. Introduction. Food security has become a
yes. Go to your full screen mode for the slides.
Pardon?
Can you change to the full screen mode, the presentation mode?
Okay.
Your slide?
Sure. Food security has become a concern of global importance, and major price spikes for staple crops such as wheat and rice. These price spikes are partly due to the brunt of plant diseases. In Malaysia, indica rice variety MR219 has been developed by Magnaporthe to increase rice production 10 to 15% higher than previous MR84 variety in 2001. Plants to survive themselves against pathogen have advanced and complicated innate immunity system, which resistant genes are parts of this innate immunity system. To date, more than 100 blast resistant genes have been identified, with 15 out of these 100 blast resistant genes have been cloned and characterized. Most of these identified genes encode nucleotide binding site leucine-rich repeat protein, except PID2 and PI21. Our objective of this study was to isolate.
Parisa, I think your slides are not in the slideshow mode.
Oh.
And they're not changing for me. I don't know if other people are seeing the slides change.
Is it okay now, bro?
No, it's still in the preview mode. It's not in slideshow.
I don't know because I already changed it to.
Somebody suggested you try clicking F5 on your laptop.
No, no problem.
Okay, one thing we can try, because sometimes it's a glitch, try leaving the meeting and rejoining and starting the presentation again. Maybe that will help. Yeah.
Okay, sure. Is it okay now, Furu?
It's still not in slideshow though. Can see the slides, but not in slideshow. So you normally have to click on the presentation mode. It's not. You're trying that and it's not opening, is it?
Presentation mode. Where can I find the item presentation mode?
At the bottom of your screen, on your right-hand side, you see the small icons. There's one for doing the presentation. Slideshow.
Yes, you are present. Already, it mentioned you are presenting.
No, you are presenting to us, but you're not presenting in the slideshow format. So inside your PowerPoint, there should be an icon at the bottom right-hand side on the PowerPoint.
Yes, yes, I did it, but I already did it, but I can show in full screen, but you cannot see it in full screen.
Okay. Someone else said try F5. F5 on your, if you have a Windows system, F5 key.
F5. Okay. Is it okay now? No. Because I can see it's on a slideshow, but you cannot see the in full screen. But I can see in full screen now.
Okay. Somebody's asking, are you working on dual screen mode? Is that the problem? Otherwise, what you could do is send your slides maybe to Dr. Nadia, maybe, or one of the SEBA staff can help.
I already sent
to Ika. Yeah, actually Ika have that slide. Maybe Ika can share the slides. Thank you, Ika.
So stop sharing and let Ika, and then you just tell her when you want the next slide. Thanks. Yeah. Otherwise, it's going to be a bit too small for people to see. Yeah. Apologies, everyone. We all know that these technical issues do happen. Okay, we're off. Okay.
but I cannot control the slides now.
You just have to tell her next slide, and she will.
Okay.
Yeah.
Okay. Okay. Next slide, please. Objective of this study was to isolate Pikh gene from resistant rice variety and to construct the overexpression vector and to and finally functional study of Pikh gene in MR219. And next, please. Problem statement. Next, please. To date, more than 25 fungal and 6 bacterial diseases reported all over the world. However, the most destructive is Magnaporthe oryzae based on ERI website. It injures plant by production of spore and penetration of infection in different parts of plants, including leaf, stalk, collar, node, and panicles. Genetic transformation has become an important means in the crop improvement strategies. The first step to carry out genetic transformation is actually having the knowledge about the sequence of the gene and information about the interest gene. However, there is not any information yet about the presence of Belacesis gene in Malaysian rice variety. Next, please. Experiment 1. Isolation of full length of Pikh gene from pH9 variety using a specific primer and RT-PCR. Experiment 2. To construct overexpression vector using gateway colony technology. Experiment 3. Tissue culture of MR219. Transferring overexpression vector containing Pikh using Agrobacterium-mediated transformation method. Confirming transformation using GFP monitoring, PCR, qPCR, RT-PCR, and HPLC. Challenging transgenic plants with fungal. Measuring fungal population with bacterial quantification technique using real-time PCR. Next, please. Material and method. To isolate Pikh gene from a pH9 variety, we needed to culture Magnaporthe oryzae on PDA and prepare spore suspension containing 3 × 10⁵ spore per ml suspension and inoculated plants. After inoculated plants and collecting the sample 31 hours after inoculation. Then RNA was extracted from the sample, converted to cDNA, and I did PCR using specific primers. Next, please. Experiment 2. To construct overexpression vector, we needed to construct entry colon and then transfer entry colon to transferring entry colon to the competent cell. And then next, please, and then confirm the insert in entry colon using colony PCR and sequencing. After that, we construct expression colon and transfer it to competent cell and confirm by colony PCR and sequencing. Next, please. And then we introduce our expression vector to Agrobacterium and confirm it by PCR colony. Next, please. To function a study of Pikh gene in MR219, we needed to do tissue culture of MR219. According to the following steps: explant sterilization, callus induction, callus proliferation. Next, please. Transfer Agrobacterium to cali, selection, transform cali on the selection media within 2 steps, shoot production, regeneration of regeneration of plantlet, root production on the MSO media, and then transfer plantlet on the Yoshida culture solution, and after that to the soil and greenhouse. Next, please. Then we check transgenic seed collected from T0 and T1 using GFP monitoring. Next, please. Analysis of transgenic plant have been done using PCR, RT-PCR, and real-time PCR. Next, please. Next. Amino acid analysis of transgenic plant have been done using HPLC. I did also some bioinformatic analysis on the sequence of gene and encoded protein by Pikh gene. Differentiation of pathogen population between inoculated transgenic and untransformed plant have been done using bacterial quantification technique. And resistance of transgenic and untransformed plant have been analyzed using after inoculation by pathogen pathotype P7-2. Next, please. Result and discussion. Isolation CDS of Pikh gene with 1206 base pair nucleotide was isolated from pH9. This gene encoded protein of 401 with 401 amino acid. Next, please. This is the sequence of gene and encoded protein. Next, please. The first figure shows the Pikh gene amplified by a specific primer flanked to the ATTB sequence. The second one shows the existence of Pikh gene in BP reaction confirmed by PCR colony. The second figure shows the presence of Pikh gene in LR reaction confirmed by PCR colony, and the last one confirmed the presence of gene in Agrobacterium. Next, please. Agrobacterium-mediated transformation showed some advantages in comparison to other gene transformation methods. It results in lower copy number of transgene insertion with easy, simple, and low-cost method. And next, please. This figure shows different stages of plant tissue, MR219 plant tissue culture and regeneration, callus induction, callus proliferation, and observing some green spot on the cali, regeneration, shoot production, and root production on the MSO media. Next, please. I did some analysis also on the fresh and dried bait of cali during 4 weeks of callus proliferation, which as you can see, the highest rate of cali have been observed within week 6, and I actually use this time to transfer Agrobacterium to the cali. Next, please. Lacking an easy, effective, and also efficient method for gene transformation in addition to regeneration can be main limitation for functional study of gene in the important cereal crops. The actual possibility of callus formation and also regeneration have been described to be varietal feature as well as successful regeneration in indica rice plant, which remains to be presents an issue for genetic manipulation via impressive approach. While more plant regeneration frequency of Japonica rice variety have been reported in compared to indica rice plant. Next, please. 4 and 9 transgenic plants were identified at the end of selection as T0 and T1 transgenic generation. Next, please. The first figure shows the presence of a Pikh gene in T1 putative MR219 transgenic plant, which is amplified by a specific primer, and second one amplified by CaMV primer. Next, please. The expression of GFP have been observed in the some seeds of T1 T1 generation. Expression of Pikh gene in transgenic and untransformed plant have been compared using RT-PCR. Next, please. Also, the expression of Pikh gene in transgenic plant have been checked and compared with untransformed, which shows upregulation of this gene in transgenic plants. The second figure shows the HPLC chromatogram of amino acid in wild type plants. Next, please. And also in transgenic plants. Next, please. Amino acid profile. The profile actually analysis of protein content of wild type and transgenic MR219 shows differences in amino acid profile of samples. Including in serine, histidine, cysteine, valine, leucine, and phenylalanine. Next, please. This figure shows the this figure shows the hydrophilicity. This figure shows the amino acid composition of a protein encoded by Pikh gene, which, according to the quantity of hydrophilic amino acid residue, we are able to predict that protein encoded by this gene is a hydrophilic protein. Actually, the knowledge about hydrophilicity and hydrophobicity of protein is actually required for predicting secondary structure and division of domain function. Next, please. This graph shows that shows and confirm that protein encoded by Pikh gene is hydrophilic protein. Next, please. Secondary structure of Pikh gene. Next, please. This is this is secondary structure of a protein encoded by Pikh gene analyzed by another software, which shows that this protein consisted of 18 sheet, 4 helix, and 21 coils. Next, please. This figure shows the subcellular localization of protein encoded by Pikh gene, which demonstrate that the amino acid sequence from 1 to 376 is a non-cytoplasmic. From 377 to 397 is a transmembrane, and from 398 to 401 is a cytoplasmic. Next, please. The first figure shows the disorder region of protein encoded by Pikh gene, which actually we didn't we haven't identified any disorder region in the protein encoded by Pikh gene, and actually we should not have any disorder region in the sequence of protein. Otherwise, this disorder region will disturb the function of protein. The second figure shows the 3D structure of protein encoded by Pikh gene. Next, please. Most of identified R gene contain a structural motif called a leucine-rich repeat, which is including a repeating pattern of 20 to 30 leucine amino acid. R genes can be subclassified into big classes. One class contains the expected genes product, encompasses of an N-terminal extracellular LRR and also a membrane anchor. And the other one class involves the R gene product, which is predicted to be cytoplasmic. Next, please. Cytoplasmic located R gene products are characterized by a conserved region, including a nucleotide binding site, nucleotide binding site, and ester C-terminal LRR domain. Next, please. The first figure shows the isolation of 20 SR DNA from Magnaporthe oryzae, which is the primary step to actually quantify the population of fungi in samples. Then the second figure is a standard curve of a gene. Next one. The first figure is amplification plot. The second one is a melting curve of Magnaporthe oryzae, which shows that this primer is specifically worked for this gene. Next, please. This photo confirmed that showed that the population of fungi after 48 hours and 72 hours of inoculation is higher in control plant compared to transgenic plants. Next, please. And also the assessment of transgenic and control plant confirmed that that the resistance of transgenic plants have been improved compared to control plants. Next, please. Bigger size of Magnaporthe oryzae population in seedling leaves of susceptible cultivar in comparison to resistant cultivar have been reported previously at 4 and 6 days after inoculation using real-time PCR quantification technique. In fact, real-time PCR is an excellent tool for Magnaporthe oryzae quantification in planta and can be applied for trustworthy evaluation of fungal pathogenicity. Next, please. Conclusion. The molecular delineation, in addition to amino acid analysis of transgenic MR219 plant, confirms some modification happened in the genome of MR219 variety. The final result of amino acid analysis in transgenic plants comparing to control plant revealed that the overexpression of Pikh gene was effective on the most amino acid contents, as the majority of variant was belong to the leucine content. Resistant analysis of T1 plants was confirmed resistant improvement of T1 plants against blast fungi. Real-time PCR quantification of Magnaporthe oryzae was confirmed the presence of higher population of fungi on the inoculated control in comparison to the transgenic plant at 48 and 72 hours after inoculation. Next, please. Hence, it can be concluded that the genetically transformed plants are able to provide unique condition which is not suitable for fungi to grow more. Next, please. I've published 7 papers from my thesis in different journals, and also presented the result in different conferences and seminars. Next, please. Next, please. Next. Thank you for your attention. And sorry for the problem.
Thanks so much. I need to apologize for technical issues. We all have faced them from time to time. It's part of living with the online world. We have this issue, especially when you're not familiar with different platforms. I know many people are using different platforms, and so maybe we're used to one and not so much to others. So this will happen from time to time. So very nice work on important disease and an important crop. I open now for, again, maybe just one question because of the timing. And again, of course, Dr. Parisa is with us in Sabah now, so we can follow up with her after the meeting if anybody is interested to contact her for further discussions. So, are there any questions? Maybe I have just one then. So is this then a single gene that can really give strong resistance in rice? And what is your comment on maybe using single genes for disease resistance?
Actually, for is a complicated system, and it's not only one gene. In the first objective of my thesis, I used 8 gene to screen this 8 gene within 17 variety, Malaysian variety, and I did PCR and real-time PCR to check their presence and also their expression. And we found out that 4 gene, when 4 gene simultaneously expressed in the variety, that varieties are resistant to shows resistant to blood disease. And we check in MR219 that this gene is the expression of this gene is constant. So that actually idea came out that we can improve its resistance by overexpressing this gene in MR219. So we try to overexpress, and we did it.
So in fact, you maybe add genes to get more enduring resistance. Do you think that is feasible?
It was visible, but it was not enough. It was not enough for totally producing actually developing a resistant variety, because so many mechanisms and mechanism are involved in the resistance. Yeah.
Do we have questions from anyone else? I don't see hands raised, but I might be missing somebody. Is anyone? If not, in the interest of time, thank you, Dr. Parisa.